Welcome everyone to the 10th and final webinar of the Science of Aging Series 2021, a joint webinar series brought to you by Inside Scientific and the American Physiological Society. For this series, we've lined up a number of webinars, all focused on the science of aging being conducted by leading researchers around the world in their relevant fields. This is Sarah McFarland from Inside Scientific, and I'm very pleased to be your host for today's event. Today's webinar is titled Cardiac Glucose Metabolism, Heart Failure in Diabetes and Aging, and will feature Dr. Dale Abel, Chair of Internal Medicine at the University of Iowa, Carver College of Medicine. In his presentation, Dale will discuss the mechanisms underlying heart failure in aging and diabetes. Before we get started, we'd just like to acknowledge our partners at the APS and the Alliance for Aging Research, and also to thank all of the sponsors for this webinar series for making it all possible. Okay, and without any further delay, I'm very pleased to welcome Dr. Dale Abel. Dale, take it away whenever you're ready.
Great. Thank you very much for the intro and for the opportunity to really participate in this really wonderful webinar series. So what I'll do today in my presentation is really focus on cardiac glucose metabolism primarily, in the context of heart failure, but also in the context of aging and diabetes. And I hope that by the end of the talk today, you will see that these really are overlapping concentric circles that have some very common themes in terms of the role of glucose metabolism in the pathophysiology of heart failure. So let me start with diabetes. And many of you think about diabetes and its complications, and the complications really drive the mortality and morbidity and suffering. And for many, many years, a lot of the focus had been on the microvascular complications of diabetes, such as retinopathy, nephropathy, and neuropathy. But importantly, it is the macrovascular complications that drive most of the suffering, and classically had been thought about as an increased risk of heart attacks and strokes. However, it's become increasingly recognized that heart failure is a bona fide diabetic cardiovascular condition. So let me just show you some epidemiological data that speaks to this. So this slide is data from the American Heart Association statistical update, and the figure was taken from a paper published by the CDC in the New England Journal of Medicine, looking at trends in certain vascular complications over a 20-year period from 2004 to 2014. The blue line that you see both on the left, which is looking at diabetes, and the right, which is looking at the general population, is ischemic heart disease. And you can see that over the 20-year period of time, there was a significant reduction in the rate of ischemic heart disease. The next line down, which is the gray line, is heart failure. And you can see that although there was a small decline in heart failure in the diabetic population, within the general population, there really has not been much of a change in the age-adjusted rates of heart failure across the population. Now, an important perspective that I think you should take away from this is that if you look at the absolute rates of either ischemic heart disease or heart failure, either in the general population on the right or the diabetics on the left, you can see a tenfold difference in the rates. So what that really tells us is that there's a residual risk that persists in individuals with diabetes. And in the bottom right, if you compare the blue bar and the red bar, you can see that ischemic heart disease and heart failure are really the two most prevalent cardiovascular complications associated with diabetes, and far exceeds stroke in the light green, lower limbs amputation in the darker green, and of course, renal failure in the purple. So if one then really looks at the relationship between glucose control and heart failure, this is an example of a meta-analysis that was published by Justin Chuba, who's North Hopkins, and it looks across multiple studies, and you can see, with the exception of one study, that there's a relationship between worsening glycemic control and incident heart failure in individuals with diabetes. In another very important population-based study, this was done by the group at the University of Glasgow in Scotland. What they did was that they looked at heart failure hospitalization data, stratifying the population by diabetes, age, and sex. So the blue bars are non-diabetic subjects, and you can see very clearly that heart failure is a disease of aging, where there's an exponential increase in heart failure hospitalizations, the inflection point being about 65 years to 70 years in men and women, respectively. But importantly, if you are diabetic, whether type 1 in green or type 2 in red, there's a leftward shift in this relationship, so that the inflection point now actually shifts almost a decade to even 15 years. So the point that I want to make from this slide is that the presence of diabetes exacerbates the relationship between aging and heart failure. Now, this is the entire population. The next slide is just removing people who were known not to have ischemic heart disease. And even if you exclude patients with ischemic heart disease, this relationship still holds true. That shifts this age relationship, heart failure hospitalizations to the left. The next epidemiological data I just want to share, again also from Justin Etruba-Chagai's work, was a survey of Medicare data across the United States, looking at nearly 370,000 heart failure patients. And what was really very insightful from this analysis was that, in fact, between 40 and 46% of patients who were hospitalized with heart failure also had diabetes. So there's a significant enrichment of diabetes in the heart failure population. It doesn't really matter whether it's heart failure with reduced ejection fraction or heart failure with preserved ejection fraction, the results remain the same. So this and other data really speaks to the notion that there's probably a cardiomyopathy of obesity and diabetes. And so it's important to really define what that means. And we believe that it describes intrinsic defects in cardiac muscle that increases the likelihood that cardiac dysfunction will develop in the context of added stressors such as ischemia or hypertrophy. Data from the atherosclerosis risk increase study that was reported by and led by Elizabeth Selvin at Hopkins really revealed very nicely that if you look at individuals across the spectrum of glycemia, if you focus on panel A, looking at individuals with hemoglobin A1Cs less than 5.7, people with prediabetes in the middle, and people with diabetes on the right. And then what she measured was the highly sensitive troponin A. And you can see that there's a progressive increase in the percentage of individuals with abnormal levels of troponin, troponin T, going from normal glycemia to hyperglycemia. So this is saying that there's a relationship between worsening glycemic control and evidence of subclinical myocardial injury. And in fact, these levels actually predict heart failure and all-cause mortality. So on the left, what was done was that there was a cutoff of 14 nanograms per liter as the abnormal level. And even in the subclinical range, less than 14 nanograms per liter, you can see that if you went from non-diabetic to diabetic, there's a progressive increase in troponin T, which of course continues once you go above the 14 nanograms per liter. But the important point is that each of these levels predicted both the subsequent diagnosis of heart failure as well as all-cause mortality in this population. So I hope to have convinced you by this that there is a strong relationship between diabetes and heart failure and cardiovascular mortality. Now the question is why? So if one knows, kind of ask the question, well, mechanistically, what might be going on? I think the concept I want you to capture, focusing in the middle of this figure, is that you have insulin resistance, obesity, diabetes that really creates this toxic metabolic milieu in which the heart lives. And then when you layer on top of that vascular complications in terms of both coronary vessels and microvessels, then you really have this synergy of both of these insults, as it were, having a negative impact on the heart. Now, in more detail, and this was a very nice cartoon in a review published by Jun Sadashima, if you start in the blood vessel and look at the abnormal sort of milieu, which is one characterized by hyperglycemia, so increased glucose, increased fatty acids, as well as increased circulation of pro-inflammatory cytokines, that there is an increased flux that characterizes diabetes of fatty acids into the heart. And there's also a relative increase in glucose availability. And so we call this either lipotoxicity or carbotoxicity. And so these carbons ultimately, through a number of mechanisms, can lead to impairment in mitochondrial metabolism, gene regulation, and also the fatty acids can accumulate within the myocyte and promulgate the generation of lipid intermediates that can then activate pathways that lead to cellular injury and cellular death. And if one even goes further into kind of molecular pathways, this cartoon is really underscoring that multiple cellular pathways have been shown to be abnormal within the heart in the context of diabetes. Another way of looking at this is that in diabetes, the heart kind of dies by a thousand flogs. Now, there's a lot of excitement recently with regards to a class of agents called SGLT2 inhibitors, which really have a significant effect to reduce the risk of heart failure in individuals with diabetes. But even with increasing use of SGLT2 inhibitors, and certainly based on the landmark cardiovascular outcomes trials that revealed this relationship between SGLT2 inhibitor usage and improvement in heart failure signs, symptoms, and hospitalizations, there remain other pathophysiological abnormalities summarized on the right side of this slide, which I think remain to be targeted and I think remain an avenue for future work. And I think the next decade will be very exciting as many of these pathways begin ultimately to be targeted. So the last point I want to make at the very high level before really spending the rest of the talk focusing on specific mechanisms by which aberrant glucose metabolism can contribute to heart failure is this concept that diabetes and heart failure share common metabolic characteristics. And this is insulin resistance. So if you look at individuals with obesity and type 2 diabetes and the metabolic syndrome, which is characterized, as we all know, by insulin resistance, at the tissue level, there are certain hallmark changes that have been described by many groups, such as ectopic lipid accumulation, for example, adipose tissue dysfunction, inflammation, and so forth. But if you look in people with heart failure without diabetes, you see the same thing. And so really, a lot of the molecular mechanisms that drive insulin resistance are shared both in heart failure and in diabetes. And so it's not difficult to begin to understand why there would be much more diabetes in a heart failure population. And also, the idea here is that when you then overlay diabetes and hypertension, in fact, what you get is an exacerbation of these underlying molecular pathologies, as it were, which I believe and others do, would then exacerbate the prevalence and the severity of heart failure in the context of diabetes. So now let me focus for this talk on specific new information as to how aberrant glucose metabolism may contribute to pathophysiology in the context of heart failure. And let me just start with this cartoon that summarizes the consequences of glucose toxicity. So when we think of glucose metabolism, we think of glucose uptake via glucose transporters, GLUT1 and GLUT4, and then the glucose undergoes glycolysis to generate pyruvate that ultimately enters the mitochondria to be oxidized. The important concept I want to convey here is that coming off the sides, as it were, of glycolysis are these branch metabolic pathways. For example, the polyol pathway, the advanced glycation end product pathway, and the hexosamine biosynthetic pathway. And all of these pathways have been implicated in ultimately leading to tissue injury. I spent a little time later in the talk talking about the hexosamine biosynthetic pathway, but just to introduce that to you now is that this pathway that branches off from fructose 6-phosphate ultimately leads to the generation of a moiety called O-GlcNac, which then leads to O-GlcNacylation, which is a post-translational modification of multiple proteins within the heart that then will have impacts on EC coupling by affecting the shuttling of calcium in and out of the sarcoplasmic reticulum, will certainly affect a number of proteins involved in gene expression regulation at the level of the nucleus, and of course will modify proteins involved in mitochondrial oxidative metabolism, which ultimately can lead to impaired energy metabolism within the mitochondria. So here's an example from our own lab. This is work led by Adam Wendy, who's now at the University of Alabama, Birmingham, recently published, where we asked the question, what does glucose do to cardiac mitochondria? And to do that, we designed a mouse model where we inducibly overexpressed the predominant glucose transporter in the heart called GLUT4. And so this was a doxycycline regulatory system, so that in the presence of doxycycline, this transgene induced about a 1.5 to 2-fold increase in the expression of GLUT4. So we had a constitutive low-level increase in glucose entry into the heart. And in fact, what we observed was this was sufficient to induce impaired mitochondrial function. And mechanistically, what we observed was that there was increased O-GlcNac modification of a number of proteins involved in oxidative metabolism within the mitochondria. And so here's one example of that shown here, which is a subunit of mitochondrial complex 1. And what I'm showing you is the 4 groups that we studied. So MG4H is that inducible transgenic mouse. STZ is streptozotocin, which is a way to induce diabetes. And so you can see that if you compare the MG4H vehicle-treated animal, and then you compare that with either the controlled diabetic or the controlled MG4H animals, you can see that there's increased O-GlcNac modifications of NDU-FA9, which is a subunit of the mitochondrial electron transport chain complex 1. This is one of many examples, but the point here is that many mitochondrial proteins are subject to O-GlcNac modifications just simply by increasing glucose delivery to the heart. The second thing that we learned from the study was that when we looked at the gene expression profiling, and so what I'm showing you here is genes in blue, which were repressed by glucose, either in the transgene alone or in the diabetic or in the combination, and in yellow, genes that were induced by glucose. And at a very high level, what we observed was that increasing glucose entry into the heart actually increased the expression of genes involved in fatty acid biosynthesis, fatty acid elongation. So stated another way is that hyperglycemia actually primes the heart to be more subject to the consequences of lipotoxicity, which is a very interesting crosstalk between those two pathways. On the other hand, the genes that were repressed were largely involved in amino acid metabolism and ketone body metabolism. So again, glucose really can have a significant effect on regulating important pathways that might ultimately govern broadly the metabolism in the heart of other substrates. Now, it's widely accepted that the failing heart is really running out of energy, and largely because of impaired mitochondrial function in those hearts. And so the concept for a long time was that heart failure was simply just energy deficit in the heart. But really, it's a little bit more complex than that, because it turns out that as the mitochondrial function becomes impaired, then the carbons have to go somewhere. And so there's increased diversion of the carbons into other pathways. So on the right, you see, for example, PPP, which is the pentose phosphate pathway, HBP, which is the hexosamine biosynthetic pathway, for example, that we hypothesized a number of years ago could also independently contribute to contractile dysfunction. And I'll show you some evidence that speaks to that. But again, just to come back to this whole notion of glycolytic overflow into these branch pathways. Again, if you start glucose and then you go down glycolysis to pyruvate, if you then go off to the right or to the left, you see these branch points from glycolysis, such as the hexosamine biosynthetic pathway to generate UDP-GlcNac, for example, or the pentose phosphate pathway, which I'm going to show you ultimately can also lead to activation of pathways that can be injurious in the context of heart failure. So let me show you some evidence from other labs that have focused on, and the other point I want to make here is that if you remember nothing else, is that think of heart failure as a block of pyruvate entry to the mitochondria. And so this is everybody's nightmare scenario where your water closet is backed up, but essentially that's kind of what happens in the heart in the context of diabetes, or of heart failure, sorry, and then you get increased flux of carbons into these alternative pathways. So let me show you some direct evidence of that from a number of studies. One of the earliest studies was done by Dr. Carlson's group And essentially what they looked at was the spontaneously hypertensive, and showed that this model that develops hypertension and heart failure had increased O-GlcNac modifications of multiple proteins in their heart. And in fact, when they looked at and in a separate model, which was essentially subjecting rodents to myocardial infarction, you can actually see that you go through a phase of hypertrophy in the middle, that's the echocardiogram, and then heart failure. And in fact, when you looked at O-GlcNac modified proteins at a global level, you can see a progressive increase in O-GlcNac modified proteins going from the hypertrophic phase to the heart failure phase following myocardial infarction. Now, Zhao Wang's group at UT Southwestern did a very nice experiment where they essentially transgenically forced flux through the hexosamine biosynthetic pathway in a transgenic mouse model. So they overexpressed the enzyme that would drive flux into the hexosamine biosynthetic pathway, and then subjected these animals in panel B to transverse aortic constriction or TAC, which is commonly used to induce heart failure in the mouse. And if you looked at in panel C, you will see very clearly that in the animals that had increased flux through this hexosamine biosynthetic pathway, they ended up after transverse aortic constriction with significantly more fibrosis. If you look at panels E and F, that's measuring cardiac hypertrophy that was also significantly increased in that particular group of animals as well. So the message here is that if you force flux through this pathway, you got much more accelerated ventricular remodeling. They then did the converse experiment where they knocked out this enzyme again inducibly within the heart, and then subjected these animals to transverse aortic constriction. And what you can actually see is that this actually was associated with a partial rescue of the hypertrophy that one would see in the context of pressure overload. So for example, if you look at panel D, you can see that measures of cardiac hypertrophy in terms of cross-section of myocytes, or heart weight to body weight, or heart weight normalized to tibial length, were attenuated when you reduce flux through this pathway. And then in panel E, F, and G, they're looking at cardiac function in these models, and you can see that the ejection fraction in panel F, or the fractional shortening in panel G, in the green bar, where the enzyme that drives this pathway was again blocked, actually was protective in this particular model. Now, so why is O-GlcNac modifications potentially injurious in the context of the heart? So what I want to really highlight from this study is really two things. Number one is that there are multiple proteins which are subject to O-GlcNac modification. So that's the G in the blue squares that are scattered throughout this figure. So for example, you can see proteins involved in calcium handling, contractile proteins are all subject to O-GlcNac modifications. As I mentioned before, mitochondrial proteins are subject to O-GlcNac modifications, as well as important signaling proteins that actually signal pathways that can lead to cell death and cell dysfunction. For example, CAM kinase, when it's constitutively activated, are also subject to O-GlcNac modifications as well. The other thing I want to introduce you to here is an enzyme called O-GlcNac transferase that transfers these O-GlcNac modifications onto various proteins. And this was then modified by another study from Mark Anderson's group at Johns Hopkins, where they either overexpressed O-GlcNac transferase or reduced its expression, and asked the question, they overexpress it or increase the expression of the enzyme that removes this O-GlcNac moiety, and ask what would happen in the heart. And again, it's a similar story to the one I just showed you, where flux was forced through this pathway. But instead, what they're doing in panel B was reducing the expression levels of O-GlcNac trans. And then, so in this study, they have reduced the expression of the enzyme that removes the O-GlcNac moiety. And in fact, what they showed in this study was that it was actually protective. So for example, if you focus on panel I, you can see, looking at fractional shortening, which is a measure, or ejection fraction, which is a measure of ventricular function, you can see that in the wild type animals, the third set of data from the left you can see a reduction in ejection fraction. But in the animals where they reduced the expression of this enzyme, then you can actually see, or I'll take it back, where they overexpress this enzyme that removes the O-GlcNac from proteins. You can see that that was cardioprotective. In contrast, when the enzyme that actually puts this O-GlcNac moiety on to proteins, or that's O-GlcNac transferase, was overexpressed in the heart. If you just quickly focused on panel N, these animals had increased mortality. You can see that the ejection fraction was significantly reduced in panel L. And so basically pushing this modification was really quite damaging to the heart. So the concept here that I want to leave you with is that increased constitutive flux to the hexosamine biosynthetic pathway is in fact damaging to the heart. And in fact, if you reduce the flux, you can actually see cardioprotection. If you increase the flux, it is sufficient to induce cardiac injury and myocardial dysfunction. So the question then is, what have we learned in studies of human hearts? Are these pathways also true? So an early study that was pioneered by Stavros Drakas at the University of Utah, and I had the privilege of collaborating with them on this study, was that we looked at patients with heart failure who were going to have an implanted device called the left ventricular assist device. And at the time of left ventricular assist device, or LVAD implantation, there is a sample which is obtained and that we then could subject to metabolomic and other analyses. And what we learned from this study was that in the failing heart, there was a mismatch, if you look at the bottom cartoon, in the relationship between glycolysis and TCA flux, such that there was a reduction of flux into the TCA cycle in the mitochondria, and so a mismatch between glycolysis and TCA cycle flux. And in fact, we had access to a second heart sample at the time of cardiac transplant in some subjects, and in those individuals who did not recover, this there's even greater exacerbation of that mismatch between glycolysis and glucose oxidation. Now, in a recent paper from the same group, they took this another layer, which is that if you do left ventricular assist device implantation, that there are going to be individuals who are going to respond and recover, and there are going to be individuals who are not going to respond and ultimately are going to go to heart transplantation. So in panel B, you will see that in the individuals who are non-responders, left ventricular ejection fraction remained low, whereas in the responders, left ventricular ejection fraction after implantation of the ventricular assist device increased quite substantially. Importantly, they looked at the expression levels of a protein called the mitochondrial pyruvate carrier that is important for pyruvate entry into mitochondria. And so what you can see here relative to the donors was that in the non-responders, the MPC proteins were lower than the donor hearts and remain unchanged in the non-responders. However, in the responders, you can see that there's a significant increase in the levels of this protein. Similarly, they looked at the phosphorylation state of pyruvate dehydrogenase. So when pyruvate dehydrogenase is phosphorylated, it reduces the flux through that enzyme. So another block, as it were, to pyruvate utilization within mitochondria. And so what in fact was observed was that relative to control hearts, PDH the phosphorylation was increased, and there was no change in the non-responders. But very dramatically, in those who responded, PDH was hyperphosphorylated, and that was reduced in those individuals who responded. So it suggests that pyruvate flux into mitochondria is an important nexus that might link the cardiac response to various stresses. So in a series of papers published in Nature Metabolism late last year, a number of groups, including our own, looked at the relationship between the levels of mitochondrial pyruvate carrier and heart failure. So in this example from the group from London, you can see that in patients with hypertrophic cardiomyopathy, or HCM, there was a reduction in the expression levels of both subunits of the mitochondrial pyruvate carrier. And also in an animal model of transverse aortic constriction, you can see that 21 days after transverse aortic constriction or attack, there's also a reduction in the levels of the mitochondrial pyruvate carrier isoform 1 and isoform 2. We also did some metabolomic profiling in another cohort of failing hearts. You can see these are end-stage hearts with very low ejection fraction and cardiac hypertrophy on the right. On the left, you can see the demographics of these patients. And we did metabolomics profiling on their cardiac samples. This is a volcano plot of that. And if you just look at the most highly induced metabolites, you can see that they're all glucose metabolites, for example, on the top right, sorbitol. So let me just show you that in a different way. This is the same data from those hearts. So we saw that there was an increase in free glucose, an increase in mannose. And why might this be important? Well, mannose can drive end-linked glycation that can lead to extracellular matrix remodeling and inflammatory signaling. We also saw an accumulation of sorbitol, and sorbitol, by depleting NADPH and glutathione, can promote oxidative stress. We also saw an increase in D-galactose and its downstream metabolites, including D-galactosamine, which is a very potent activation of the ROS-H rate signaling pathway and a promoter of inflammation. So again, underscoring the fact that there's increased flux through these other glucose pathways, which we believe can then activate signaling pathways that can promote myocardial injury. So to address this directly, Yuan Zhang in the lab and Eric Weatherford decided to either look at the impact of putting more glucose in through oversuppressing GLUT1, or to block the ability of the mitochondria to metabolize pyruvate by inactivating the mitochondrial pyruvate carrier. And so the hypothesis for the mitochondrial pyruvate carrier work, which is what I'll describe right now, is that we would basically create, in a mouse model, this glycolytic glucose oxidation mismatch. And the question would then be, mechanistically, would that lead to ventricular remodeling? So here's the mouse model. We knocked out the subunit 1 of MPC, and MPC2 was also degraded because they live in a complex, and so loss of MPC1 leads to degradation of MPC2 as well. You can see in the right that there's a significant reduction in mitochondrial pyruvate uptake, and also pyruvate-supported mitochondrial respiration. The black lines in the bottom are controls. The red lines in the bottom are the knockout animals. We also saw that increased O-GlcNac modification of many proteins. So on the Western blot, in panel G is showing an O-GlcNac blot. You can see in panel H the quantification of those blots, that there's an increase in O-GlcNac modification. We also saw that there was glycogen accumulation as well in the hearts that was more evident in animals that were in the fed state. So these animals spontaneously developed heart failure, and they had an age-dependent cardiomyopathy. They essentially developed heart failure with or pathological ventricular hypertrophy at 8 weeks, and then dilated cardiomyopathy at 14 weeks and beyond. You can see that the ejection fractions progressively reduced, and then in the bottom right, you can see that these animals had increased mortality. So we then said, well, is this just simply because these animals just are missing an important substrate, namely glucose? So we did what we thought was kind of a simple experiment, which was just to try to compensate for this by giving them another substrate. And we did a couple of experiments. One, we either put the animals on a ketogenic diet to increase the availability of ketones, which in the mouse model is a high-fat, low-carbohydrate diet. And we also put them on a high-fat diet. And what we basically showed that we could prevent the heart failure by starting the diet shortly after weaning, before at the point when the hearts were structurally normal, or we could reverse the hypertrophy at the stage of compensated ventricular hypertrophy, or even reverse the heart failure if these diets at a later age. So let me show you an example of that. So these are mice that were in heart failure, and when we put them on the ketogenic diet, you can see very clearly that there was a regression of ventricular hypertrophy and a normalization of ejection fraction. And you can see at the end of the experiments, when we measured the heart weights, that there was also a complete normalization of heart weights normalized to tibial lengths. Importantly, though, this was only true as long as the animals remained on the diet, because if we then took these animals who were rescued and then either randomized them back to normal chow or to the ketogenic diet, in fact, what we observed was that when we put animals back on the normal chow, again, you could see in panel B that the hypertrophy reoccurred, and that and panel D as well, and the heart failure also relapsed. So you can see that there is this switch that that occurs that we were uncovering when we had the animals on the ketogenic diet. So the question was it the ketones or something else? So to address that, we did metabolomics profiling. This is a very busy slide, so I just have it sit here for a while so you can digest it. So there are 4 groups here: the animals, wild animals on a controlled diet, knockout animals on a controlled diet, and then wild type animals on a keto diet, and then the knockout animals on a keto diet. And maybe if you just focus, for example, and these are long-chain acyl carnitines that we're profiling in these hearts. So if you maybe just focus on C16O, you can actually see that the knockout animals on normal chow actually had higher levels of acyl carnitines, even at baseline. When you put wild-type animals on this diet, they also accumulate acyl carnitines, but the knockouts actually don't accumulate anymore. And so the way that we interpreted these data was that these hearts were already adapted in a way that they probably were using more fatty acids at baseline and already were at sort of their capacity, as it were. And so the question then is, what about glucose metabolism in these hearts? If you focus over on the right, looking at pyruvate and lactate, what you can see very clearly is that when the MPC mutant animals were placed on the ketogenic diet, you can see that the accumulation of pyruvate and lactate that occurs in normal chow was completely prevented. So it's the other way, and you can maybe just look over on the far left at glucose, that these, we believe that we essentially shut off glycolysis in these hearts by putting them on this diet. And in fact, what that was correlating with was an improvement in energy metabolism in these hearts. You can see that acetyl-CoA levels went up. You can see that ATP levels went up in these hearts. And this correlated with a reduction in O-GlcNac modifications, a reduction in glucose. So what I think we were really doing on the ketogenic diet was really suppressing flux through glycolysis, suppressing flux through these other pathways that I described, which then correlated with ventricular remodeling. So it wasn't just simply about substrates. And the other experiment that we did that I think kind of convinced us that that was probably also the case was we actually subjected these animals to transverse aortic constriction. And so if you take these pyruvate deficient hearts and you do transverse aortic constriction, in fact, what we see if you look at the red lines in the animals on normal chow is early mortality. Now, in the top experiment, we also had another group of animals that we put on the ketogenic diet exactly at the time of transverse aortic constriction, and this did not change the phenotype in terms of the premature mortality. However, if we treated these animals on the ketogenic diet 3 weeks prior to TAC, so we essentially shut off glycolysis in these hearts and then repeated the surgery, we saw a different effect. So here are animals on normal chow. Again, you can see a very dramatic reduction in ejection fraction after TAC in the mutant animals. But when we pre-treated them with the ketogenic diet for 3 weeks before, you can see that these hearts now ultimately became resilient. So what I think this is telling us is that the left ventricular remodeling is not solely dependent on alternative substrate availability, but a broader remodeling of glycolytic metabolic pathways that promote cardiac protection. And this is true in human hearts as well. In a paper published in Circulation, we observed that in the same human cohorts that were subjected to ventricular assist device implantation, that in fact, if we looked closely at those who recovered, we saw a remodeling of glucose metabolic pathways in their hearts in a way that we would predict would promote cardio protection. And then the final kind of data slide in the heart is really a very interesting experiment that was again done by Stavros Drakos' group, where they induced heart failure by catecholamine excess in a rodent model. And then what they observed was, again, there was reduced flux of glucose carbons into the mitochondria through the pyruvate carrier, increased generation of lactate, and did a clever experiment where they inhibited the export of lactate by treating these animals with an MCT4 or a monocarboxylate transporter 4 inhibitor to force these carbons back through them into the mitochondria. And this correlated with evidence consistent with reverse remodeling in this particular context. So what hopefully I've been able to to make the case for is that, we believe that when there is a mismatch between glycolysis and glucose oxidation, that there's an accumulation, as occurs in heart failure, as occurs in diabetes, that there is an accumulation of these glycolytic intermediates that can drive ventricular remodeling. Now, I think what's really interesting is that this phenomenon is probably not just only, restricted to the heart, because if one thinks about cancer metabolism, there are actually some important parallels. So as you know, in cancer cells, there's also this induction of glycolytic metabolism, and with flux through the PPP or the pentose phosphate pathway potentially being very important for proliferative synthesis in these cells, as well as lactate, which has been shown to drive metastases, for example. And it turns out that there are important checkpoints that have been well described in the cancer literature that drive this phenomenon. For example, PFK1, which actually has also been studied in the heart a long time ago by Rang Tian, who showed that in a heart failure rodent model that many inhibitors of PFK1 were actually accumulating within the hearts of those animals. Similarly, another important checkpoint in cancer, which is pyruvate kinase or PKM2, Heinrich Teitmeyer showed very nicely that that pathway is also activated within the failing hearts. MPC has also been implicated in many cancers now, and I already showed you data from in our hands that is the case. And although I won't show you the primary data for this, we and others have also shown that novel oncometabolites, for example, 2-hydroxyglutarate, is also significantly induced in the context of glycolysis, glucose oxidation mismatch. So really, we think that heart failure, cancer, common disorders of aging perhaps share a common metabolic soil, in the sense that in heart failure, the glycolytic intermediates are really driving a process of ventricular remodeling, whereas in cancer, these pathways are driving cellular proliferation. So I hope to have at least, sort of tweaked your interest in thinking about aberrant glucose metabolism really playing an important role in two very important disorders of aging, specifically heart failure, and I just at least teased you a little bit with this being true in cancer as well. I want to acknowledge the members of my lab. This is our COVID selfie picture, and particularly Yuan Zhang in the bottom right, who really led the studies with the mitochondrial pyruvate carrier. And of course, importantly, to acknowledge many collaborators who contributed to the outcome of these studies, and of course, our various funding sources. So again, thank you very much for your attention, and I hope that this presentation has stimulated some questions.
All right, so the first question here for you is, for patients with heart failure, is it recommended to follow a ketogenic diet?
Yeah, so that's a good question. So that is not currently a recommendation. Having said that, there are a number of trials of increasing ketone availability to the heart in heart failure. One of the challenges, of course, with the ketogenic diet is that it's a high-fat diet that's either restricted in protein or carbohydrates. I don't think we fully understand enough as to the potential negative consequences of that sort of dietary composition. Groups are also looking at just ways of supplementing ketones. Ketones are not very palatable, so that does create a little bit of a challenge. Although there are companies now that are actually making ketone preparations that will reliably increase ketone levels. So there are trials ongoing now in the heart failure population to determine if, in fact, these dietary or pharmacological approaches to increase ketones will ultimately be beneficial. Certainly, the data from large animal studies are encouraging, and so I think we will have some more clarity about that in the next few months to years.
Great. Okay, the next question here, and a couple people have asked about the mitochondrial function stuff, so I'll kind of group a couple of them together, hopefully. This question is, has anyone looked at the mitochondrial function from the failing hearts, quote unquote failing hearts, in the knockout mice versus the non or wild type mice, and specifically with isolated mitochondria from cardiac muscle?
Yeah, so I think the questioner is asking about the pyruvate carrier knockouts. And so the answer is yes. If you look in our Nature Metabolism publication, we actually did mitochondrial respirometry in these hearts, and very importantly, and also looked at ultrastructure, etc. And I think the important point here is that in those mutant animals, the only defect that we could discern was an impairment in oxygen consumption when pyruvate was the mitochondrial substrate. If we use other substrates like succinate or palmitoyl, carnitine, for example, we saw no respiratory phenotype. And when we looked ultrastructurally on these mitochondria, we saw no ultrastructural defects. So essentially, the ventricular remodeling and heart failure that we are seeing in this particular model is not really a function of an intrinsic defect in the mitochondria per se, but really we believe just a change in the traffic patterns of glucose-derived carbons to essentially flood the glycolytic pathway with carbons that then had to go off into these side branch pathways. So to really kind of answer the question, we think that the mitochondria are intrinsically healthy. Another thing that you'll see when you read our paper is that, in fact, early on in these animals, there was an adaptation of these animals where, at least in the short term, there were an MPC-independent pathway of pyruvate entry that lasted for a while, but wasn't enough to sustain normal cardiac structure and function as these animals aged.
Okay, fantastic. This next question here is, well, first it's a thank you, Dr. Abel. Then the actual question is, high-fat diets also promote cardiac hypertrophy, but you showed that a ketogenic diet improves the cardiac phenotype. Is the high-fat diet somehow reducing the glucose flux, but ketogenic diet isn't, or is it a different mechanism?
Yeah, so again, a great question, and I think there are multiple levels of complexity here. So when you put animals on a high-fat diet, they become insulin resistant and hyperinsulinemic. They become a little bit volume overloaded because insulin actually will increase sodium reuptake from the kidney. They become a little hypertensive as well. So all of these, so the hypertension, the volume overload, can also drive cardiac hypertrophy. And then, of course, we have shown in other studies that hyperinsulinemia can also promote pro-hypertrophic signaling pathways within the heart. So I think there are multiple mechanisms by which the high-fat diet feeding can drive cardiac hypertrophy in an otherwise normal host. I think that what we kind of did in the pyruvate knockout studies was essentially we used, whether it was high-fat diet feeding or a ketogenic diet, and I think it's important to emphasize that what I showed you in the talk here for the ketogenic diet, we also saw the same effect by putting animals on a non-ketogenic high-fat diet. So wasn't anything necessarily magical about the ketones per se. It was just having a manipulation that just kind of suppressed glycolysis sufficiently to kind of, as it were, drain the swamp of all these carbons that were kind of, filtering off from glycolysis, and we were able to show that correlated with reverse remodeling. So I hope that has addressed that question.
Yeah, I think it did. Thank you for that. I love the drain the swamp analogy there too. Okay, so this next question here is, can you say a word about glucose-6-phosphate and its role at the crossroads of glucose metabolism?
Yeah, so again, not fully sure, exactly what the question is, but I'm just gonna quickly, if I can, just gonna scroll back to that glycolysis slide here. That might help to address this. I'm just scrolling back to the cartoon of glycolysis here. I think it's here we go. Okay, so that's slide 30. Can we go back to slide 30? I think. Oh, absolutely. I think I need to pull it up. Yeah, that's that one. Yeah. So I think the question is glucose 6-phosphate. So you can see where glucose 6-phosphate sits within the glycolytic pathway. So an important so G6P is an important precursor, for example, into the pentose phosphate pathway, as you can see here in this figure. And clearly, one of the things that we have seen in the MPC mutants, but also in human heart failure, for example, one of the most highly induced metabolites that we saw in the MPC mutant animals was this metabolite that you see if you go from glucose 6-phosphate to 6-phosphogluconate to ribose 5-phosphate is pseudoheptadose 7-phosphate. And that was quite induced. That was also true in humans with heart failure as well. The other point I think about G6P is that when G6P is converted to 6-phosphogluconate through G6P dehydrogenase, there is consumption of NADPH. So this pathway also plays a very important role in redox regulation within the heart. And so I think that's another important aspect that we didn't know about this study, but I think certainly potentially could be implicated again in this pathway. And finally, the pentose phosphate pathway, again, which is downstream of glucose 6-phosphate, ultimately can also lead to the generation of nucleotide precursors, which are, as again, important building blocks for potentially important in cellular proliferation and potentially also in cardiomyocyte hypertrophy. So I do think that it is an important nexus involved in pathological hypertrophy. There's very nice work from Brad Hill at the University of Louisville was also shown that it's an important switch as well in comparing the metabolic adaptations to pathological hypertrophy, which is what I really mainly talked about, versus physiologic hypertrophy, which occurs, for example, in the context of exercise.
Fantastic. Okay. We've got tons more questions here. Keep them coming, guys. And we don't have a ton of time left, so I'll just do as many as I can. This question is, can you please address or touch on the role of SGLT1? I believe that is the sodium glucose cotransporter 1, specifically in the context of diabetes and heart failure.
Mm-hmm. Very good question. So this is not my work, but work by others that have suggested that in the context of, so let's just back up. So if one looks at glucose entry into the heart, there's the glucose transporters, group 1, group 4, but there's also these SGLT1, which essentially are sodium glucose co-transporters. And there's evidence in diabetes that there's induction of SGLT1 in the heart, and that basically is correlated with activation of certain growth-promoting signaling pathways downstream of MAP kinase, but also pathways that might be involved in calcium homeostasis within the myocyte. So I do believe that there is emerging evidence that induction of SGLT1 potentially could also be playing a role in cardiac dysfunction in the context of diabetes.
Okay. Fantastic. We've got a couple questions here that are addressing the same thing, so I'm going to try and combine them for you. Essentially, people are wondering if these same dysfunctions in glucose metabolisms in cardiac muscle with the same disorders can be observed in skeletal muscle as well, especially with things like sarcopenia. And do you know if there's any mechanism that can underlie both cardiac muscle and skeletal muscle?
Sure. great question. So I don't think they're necessarily parallel. So Eric Taylor here in the University of Iowa has knocked out MPC1 in skeletal muscle. And it's a complex phenotype, and this was actually published in eLife a year and a half ago. That essentially what then happens when you block that pathway is that there is actually increased efflux of carbons to the liver, kind of like a reverse of the Cori cycle, as it were, which then has a systemic metabolic effect. So I think that skeletal muscle is a little bit different in that because there's so much of it, and that it's really a source of carbons that actually get exported to the liver, that the phenotype of the skeletal muscle MPC1 mouse really is more one of redirection of carbons to the liver, as opposed to primarily driving sarcopenia. That said, I don't think Eric really focused a lot on muscle mass in that particular study. So and I don't know if people have really significantly or systematically looked in any of the many models of sarcopenia to then ask the question, once you develop sarcopenia, do you actually get, is there evidence of mismatch between glycolysis and glucose oxidation? So certainly I think it's ripe for future study.
Great. All right, another question here. Are you aware of any stable isotope tracing experiments in these models? Specifically, do you know how the levels of TCA cycle intermediates play out?
Yeah, so if I refer you to our Nature Metabolism paper, we did do stable isotope tracing studies in isolated perfused hearts, and we're a little initially confused because I said earlier, it looked like there was a MPC independent pathway that was kind of keeping the TCA at least initially filled up in the compensated stage. But however, as these animals aged, there was clearly a reduction in the TCA intermediate pool, at least in the heart. Eric Taylor has done elegant tracing studies looking at in animals with MPC deficiency in skeletal muscle and liver, and I would certainly encourage you to go look at those papers. The liver paper was in Metabolism, and as I said, the skeletal muscle paper was in eLife.
Amazing, great. And we can always post the links to those papers in the resources section for the on-demand viewers as well. Another question here, what drives protein degradation in the reverse remodeling?
Oh, I don't know. I don't know the answer to that question, because when you I mean, I think you're really asking, you're assuming that the reverse remodeling is associated with protein degradation, but remember it's probably also associated with some changes within the matrix as well. But we don't really know what drives that, and we haven't yet done kind of flux studies to kind of see what's going on. What I can tell you though is that in a very nice study that was published in Science by Zolta and his group at the University of Pennsylvania, what is very clear is that there's tremendous amino acid flux in and out of the heart. So this was a study he did in humans where he categorized both the right and the left side and essentially looked at metabolite flux across the heart. And I think one of the unexpected but important findings of that work was that there's tremendous protein turnover and amino acid turnover in the heart, even of relatively healthy people. So, but if the question is mechanistically what drives that, I don't know. And I think that really opens up, and again, many interesting questions. I mean, we don't think in our hands was autophagy because we looked at that, but ultimately, what's driving the breakdown and recycling of these amino acids, I think, is still a relatively open question.